So the cell goes through a series of stages of growth in its life. An interphase is when DNA replication occurs, resulting in 2 copies of each chromosome and letting us move forward to mitosis. The first stage that we start with is prophase, and in prophase the chromosomes start to condense and the nuclear envelope, which is there, begins to break down. The next stage is metaphase, where microtubules attach to each of the chromosomes, and then the chromosomes begin to line up at the metaphase plate in the center of the cell. In anaphase, the microtubules begin to push apart towards the poles, and they pull the chromosomes apart towards the 2 opposite poles of the cells. In telophase, the microtubules break down, and then the chromosomes, they start to decondense again, and the nuclear membrane begins to reform. This is also when the cytoplasm begins to cleave in 2, getting ready for the splitting of the cell into 2. This is also when cytokinesis occurs, dividing the cytoplasm apart to form 2 new daughter cells. So this is the end result of mitosis, which is 2 new diploid daughter cells, each with 2 copies of each chromosome.
So DNA replication occurs during interphase, and as you can see there, the DNA represented by the blue and the red, and in the interphase they replicate, resulting in 2 copies of each chromosome. So the first phase of meiosis 1 is prophase 1. And in prophase 1, the chromosomes begin to condense. But as well as this, crossing over occurs of the alleles on different chromosomes, and you can see that by the mix of colors between blue and red of each chromosome. And and this allows for more variation within them. Also shown here is the nucleus surrounding. The next phase is metaphase 1, and in metaphase 1, the chromosome pairs, in our case, the top two are one pair, and the bottom two are another pair. So in metaphase 1, they begin to line up at the metaphase plate, as shown here. So they go together in the middle and line up. And then the microtubules, or tubules, attached to each chromosome, as so. The next phase is anaphase 1, and in this phase, the microtubules push apart towards the poles and pull the chromosome pairs apart. So as shown here, the chromosome pairs are now going to be pulled apart from each other, but the two sister chromatodes of each chromosome will stay together. The next phase is telophase 1, and in this phase, the microtubules will break down, but the chromosomes decondense and the nuclear membrane will reform. So as shown by this drawing, the cell is beginning to split in order to create two new cells. And this is where cytokinesis occurs, which divides the cytoplasm to form two diploid cells. So now as the result, we have two diploid cells, each with their own chromosomes from the last part. And now with these, the process can occur all over again. So now we move into meiosis 2, and from now on everything happens twice because we are working with two cells. So this first phase is prophase 2, and within it the chromosomes condense and the nuclear membrane surrounding them begins to break down. And drawn here are two different pairs in two different cells of chromosomes that are mixed up. So yeah, here is the nuclear membrane. We now move into metaphase 2. Similar to mitosis, this is where the chromosomes line up at the metaphase plate. So we made a little error on the top one. They are actually lined up, not side by side, but which we'll fix in a second. But top to bottom, they line up at the metaphase plate. And from there, the microtubules will attach again to each chromosome in order to prepare the split apart. So next we move into anaphase 2. And here, the microtubules will pull each chromosome apart in order to separate the sister chromatids to the opposite ends of the cell. So as you can see, compared to metaphase 2, the chromosomes are being pulled apart, pulled in half, so that there's a sister chromatid on each side of the cell. And finally, we move into telophase 2. Within telophase 2, the microtubules will break down. The chromatids decondense. As shown here, they're not as structured or condensed as before. And in addition, the nuclear envelope reforms. And shown by the indents within the middle of the cell, it is preparing to split again. And yeah, so then cytokinesis occurs twice, one for each cell, which results then in 4 haploid daughter cells. So yeah, as shown here, we now have 4 haploid daughter cells, and each with only 1 copy of each chromosome. And that is meiosis.